We have started our biochemistry and we have seen biochemistry of carbohydrates, biochemistry of lipids. We have also seen amino acids and proteins. So out of that, we have also seen vitamins from biochemistry. So carbohydrate, proteins, lipids, vitamins, amino acids. So these chapters are completed. So from the amino acids, I'm going to take I will make one more chart and I will present in front of you because I think the metabolism of individual amino acid is remaining, and as I told you, this metabolism chapter is a very lengthy chapter and it is a very vast chapter. So a metabolism of seeing each and every single thing from metabolism is not possible to cover. So I have taken uh, I have taken whatever is important. So again, I would like to take one more session for vitamins and enzymes. So whatever remaining points are there, I will cover in upcoming lecture. But today we will see our new chapter that is known as a biochemistry of enzyme. So biochemistry of enzyme. This chapter again is important. You can see there was a question in GPAT 2020 about the classification of enzyme. Okay, then lot of time they used to ask question on the competitive inhibition, non-competitive inhibition. Most of the time they used to ask question like enzymes and their cofactors, enzymes and their vitamin cofactors, etc, etc. Okay, so this chapter is again very important for GPAT Niper DI Pharmacist. I hope I am audible to you clearly, and are you able to see the screen properly? Screen display properly. So here we'll see. First of all, what do you mean by enzyme? So enzyme means they are nothing but the biocatalyst. So what do you mean by enzymes? The term simple enzyme. Enzyme means they are nothing but the biological catalyst. What is meant by catalyst? Catalyst is any substance that will not take part in the reaction, chemical reaction, but definitely it will increase the velocity of reaction. It will increase the rate of reaction. Okay, like a perfect example of this enzyme is nothing but a role of teacher in the life of students. So a teacher is a catalyst for a student because most of the time students they find difficulty in the books, so they didn't understand the chapter. Okay, if they started reading, so what is the role of teacher? So teacher used to study that chapter, teacher used to explain that chapter in a simple language to the students, right? And that is the catalyst, so that student will understand it faster. You can correlate this enzyme role of enzyme is exactly like a role of teacher in the student's life. Okay, so enzymes are nothing but the biocatalyst and they are protein in nature. Enzymes are nothing but the biocatalyst and they are a protein in nature. Okay, biocatalyst. Okay, they will increase the rate of reaction without undergoing utilized in the reaction. वो enzymes क्या करतील? Enzymes क्या करेंगे? वो reaction rate बढ़ाएंगे. They will increase the reaction rate, but they will not get consumed in the reaction. ये reaction में वो rate बढ़ाएंगे, उसका velocity बढ़ाएंगे, production product formation का rate बढ़ाएंगे, लेकिन वो खुद utilize नहीं होंगे enzymes. ठीक है? So enzyme so again, enzymes are the biocatalyst. Okay, they are a protein in nature. They have a very specific action. They are having rapid action and they are having accurate action. Okay, they are very specific, rapid and accurate in their action. Okay, they will increase the velocity of reaction without itself undergoing any change in the overall reaction. तो reaction में कुछ में कोई change नहीं होगा enzyme में. Okay, so remember they are the biocatalyst. They are the protein in nature. They are specific, rapid, accurate in action. Okay, they increases the velocity or rate of reaction or rate of product formation without themselves undergoing any change in the overall reaction. Now enzymes, what they did? There was a question in GPAT 2022. Okay, simple question was there. So enzymes increases the rate of reaction or enzymes increases the rate of reaction or enzyme increases the formation of product in the enzyme catalyzed reaction by option में नीचे था option 1, option 2, option 3, option 4 by increasing the activation energy, by lowering the activation energy. Okay, by by increasing the collision between your substrate and enzyme, and there was one more option. Okay, so how enzymes will increases the rate of reaction. How enzyme will increase the rate of reaction? So enzymes lowers the activation energy. What is the activation energy? यहाँ पे activation energy होता क्या है? So activation energy is the minimum energy required to cause a collision between two molecules. अगर दो मॉलिक्यूल अगर एक में के साथ में इंटरेक्ट नहीं होंगे, एक में के साथ में कोलाइड नहीं होंगे, एक में के साथ में कांटेक्ट में नहीं आएंगे, तब तक उनके बीच में कोई रिएक्शन होने का चांसेस नहीं. So enzyme what they does, they decreases the activation energy between reactant and okay between the reactant. Okay, they decreases the activation energy. This was a question. This was a question in GPAT 2022 recently. So enzymes lowers or they decreases or they decreases the activation energy required for the enzyme catalyzed reaction. This was question in GPAT 2022. Okay, so simple, just within a one or two lines there was a question. What is meant by enzyme? So I told you. So who coined the term enzyme, or who was first time isolated the enzyme? There is one scientist known as a Buckner. Buckner in 1883. He was isolated one principle known as a zymase. One principle known as a zymase. And what is zymase? It is nothing but the enzyme. Zymase, this enzyme was able to, this zymase was later on found to be protein in the structure. So this Buckner in 1883 isolated this active principle known as zymase, which was later on found that it was a protein in nature. Now again James Summer first achieved the isolation and crystallization of another enzyme known as a urease. What is the role of zymase? So zymase will convert sugar into alcohol. Zymase का रोल क्या होता है कि zymase अगर ग्लूकोज के अंदर zymate डाल दिया वो अल्कोहल बनाएगा उसका. So zymase will convert sugar into alcohol. Okay. Another enzyme which was first time isolated in a crystal form, that is a urease from jug bean. जग फ्रूट जो होता है उसका जो बीज होता है, जग फ्रूट का बीज, उससे वो एंजाइम यूरेज आइसोलेट किया था. सो एंजाइम क्या होता है? एंजाइम इज नथिंग बट द बायोलॉजिकल कैटलिस्ट. They are nothing but the biological catalyst, and without this enzyme life is not possible. Why? Because if you try to digest something, है ना, अगर आपको प्रोटीन का डाइजेशन करना है, if you want to hydrolyze the protein, okay, in the laboratory, it will take around few few days for a complete hydrolysis of protein using acid. आपने strong acid भी यूज किया तो भी कोई प्रोटीन को पूरा डाइजेस्ट करने के लिए, पूरा हाइड्रोलाइज करने के लिए इंडिविजुअल अमीनो एसिड रिलीज करने के लिए it will take around 2 days or 3 days. It will take around 2 or 3 days for a complete hydrolysis of protein in laboratory by using acid or alkali. Now same thing is done in your body. Same thing is done in your body within a few hours. Within a few hours. So how it is possible? So it is only possible because of the enzymes present in your body. So you are eating biryani, you are eating non-veg food, you are eating chicken, mutton and all that. So what are they? They are nothing but the protein in nature. Now that is digested in your GIT. So in the your GIT there is the release of enzymes. You have pepsin, you have trypsin, you have chymotrypsin, you have proteases, you have glycase, you have lipase, you have sucrase, etc., etc. So which will digest particular type of, or which will hydrolyze particular type of chemicals. So sucrase will hydrolyze sucrose, glycase like glucose, lipase will break down the lipids, like that. That's how protease will down to the protein, trypsin, chymotrypsin, right? They digest the proteins, so they will hydrolyze the protein, and there is release of there is there is a release of oligopeptides from polypeptides. There is a release of okay, simple peptides from oligopeptides. There is a release of finally amino acids from the peptide or protein. So this process will take around 1 to 2 days in a strong acid in the laboratory. But same thing is done in your body within a few hours, and that is only possible because of the enzymes. So that's why enzymes, they will decrease the activation energy and they will increase the rate of reaction. They will increase the velocity of reaction. They will increase the velocity of reaction. Right. So Buckner in 1883 first time isolated a zymase enzyme which can convert glucose into alcohol. Okay. Then James Surner first he achieved the isolation and crystallization of urease enzyme from jugwin. Right. I hope I am audible to you clearly. Now enzyme, yes, Am I audible? Now, enzymes may be either intracellular enzymes or extracellular enzyme. So intracellular enzyme means what? So intracellular enzyme is they are produced in the cell, inside cell, and they are released inside the cell. है ना? जो एंजाइम सेल के अंदर ही बनता है और उसका अंदर ही यूज होता है ना? वो अंदर वाला है, बाहर वाला नहीं. अंदर वाला एंजाइम क्या है? इंट्रासेलुलर एंजाइम्स. एंजाइम्स में बी आइदर इंट्रासेलुलर or extracellular. Enzymes may be either intracellular or extracellular. So intracellular enzymes which are produced in the cell, and utilized in the cell for chemical reaction. They are the intracellular enzyme. And extracellular enzyme is they are produced in the cell, but they are released outside the cell, and they have their function over there. Right? Extracellular enzyme. So all digestive enzymes, this is trypsin, chymotrypsin, all these are the digestive enzymes which are extracellular enzymes. Now these enzymes may be given a name, different name, right? Right. For example, so glucase, amylase, okay, such kind of names they are initially they were given the name like this, depending upon the discovery, the depending upon the product on which they act. Okay, but later on trivial names of enzymes was given. Okay, like lipase means there is some idea it will break down the lipid. Proteases, there is some idea it will break down the protease. Ase, okay, they are having suffix ase means they are the breaking down, and they will break down something. So protease will break down protein, lipase will break down lipids. Nucleages means they will break down the nucleic acid. Okay, but now after 1886, okay, after 1886, so International Union of Applied Biochemistry, okay, International Union of Biochemistry, they have designated particular names for the enzymes, है ना? उसके बाद में बायोकेमिस्ट्री के आप जो होता है इंटरनेशनल यूनियन ऑफ बायोकेमिस्ट्री, they have given the names to the enzymes depending upon their function. They have classified enzymes in a six part. So we are going to see that classification also, so don't worry. So enzymes may be classified as intracellular, extracellular. It may be they may be given a name like they may be named depending upon their discovery. They may be named on their trivial names where they act. Enzyme is a protein in nature along with another part. Okay. Now active enzyme is known as a hollow enzyme. Active enzyme is known as a hollow enzyme. Remember, this is very important question. There was already question on this. Hollow enzyme is an active enzyme. ये क्या होता है? Active enzyme. Hollow enzyme is an active enzyme, right? Now it is composed of a two part. First part is the apoenzyme, which is known as a protein part of enzyme. So apoenzyme, and second part is nothing but the coenzyme. Coenzyme is nothing but the non-protein part. Coenzyme क्या होता है? Non-protein part. So okay, non-protein part. Okay, now active enzyme known as a hollow enzyme, and it is composed of a two part. So first part is a apoenzyme and second part is a coenzyme. Apoenzyme क्या होता है? Apoenzyme is nothing but the protein part of your enzyme. Okay, and coenzyme is what? So in the coenzyme it is a non-protein part. Now what is present in the coenzyme? So coenzyme में क्या होता है फिर? So there might be some metal, there might be some there might be some cofactor, there might be some, okay, non-protein another group, so known as a prosthetic group also sometimes. Okay, so coenzyme is a non-protein part. Remember, apoenzyme is a protein part, and active enzyme is known as a hollow enzyme, and which is composed of apoenzyme and coenzyme. इसके ऊपर भी क्वेश्चन आ चुका है, ठीक है, ऑलरेडी क्वेश्चन आ चुका है. नाउ द टर्म prosthetic group. The term prosthetic group is sometimes used. Okay, prosthetic group. The term sometimes prosthetic group is used. It is non-protein part which is covalently bound with the apoenzyme. You know, most of the time क्या होता है कि enzyme, hollow enzyme is composed of apoenzyme and coenzyme. But most of the time coenzyme is a part which is a non-covalently bound with the apoenzyme. But if something is bind covalently with the apoenzyme, and that is known as a prosthetic group. So remember, hollow enzyme is an active enzyme and composed of a apoenzyme which is known as a protein part, and coenzyme which is known as a non-protein part. Now if coenzyme is attached to a apoenzyme by covalent, so it is known as a, okay, It is known as a prosthetic group, okay, which binds with the upper enzyme, right? Are you getting or not? समझ रहे हैं आपको? So functional unit is known as a hollow enzyme. Now there are multiple forms of same enzyme. It is known as a isoenzyme. For example, phosphodiesterase 1, phosphodiesterase 2, phosphodiesterase 3, 4, 5. So there are 5 isoenzymes, right? So multiple forms of same enzyme is known as a isoenzymes. So definitely these forms they will have different location, they will have different action, okay, they will have different function. Functional unit of your enzyme is known as a hollow enzyme, and enzyme may be a different form known as a isoenzymes, right? And known as the isoenzymes. Now next one is nothing but the, okay, now if in the enzyme, if only enzyme is made from a single polypeptide, अगर एंजाइम में एक ही प्रोटीन का है, एक ही प्रोटीन है, एक ही सिंगल, if enzyme is made from a single peptide known as a monomeric enzyme. Now, okay, monomeric enzyme, oligomeric enzyme, and polymeric enzyme. There are three terms. Monomeric means the enzyme is made from single polypeptide. Example is nothing but the ribonucleases and trypsin. So they are made from a single peptide known as a monomeric enzyme. And oligomeric enzyme मतलब क्या? So they are made from few peptides. For example, more than 5 to 10, etc. etc. So example is a lactate dehydrogenase and aspartate transcarboxymalase. So these enzymes they are known as oligomeric enzyme. They are made up from a few peptides. अगर सिंगल पेप्टाइड से एंजाइम बनाए तो उसको बोलेंगे मोनोमेरिक एंजाइम. अगर 5 से लेकर 10 तक बना है पेप्टाइड है उसमें तो उसको बोलेंगे ओलिगोमेरिक एंजाइम. अगर बहुत ज्यादा है तो उसको बोलेंगे पॉलीमेरिक एंजाइम. एंजाइम्स में भी कॉल्ड एज अ multi enzyme complexes or polymeric or monomeric. Multi enzyme complexes means what? So it is composed of 2-3 enzymes, okay? So for example pyruvate dehydrogenase, इसमें 2-3 एंजाइम लगता है. फैटी एसिड सिंथेसेस, okay? So that is known as a multi-enzyme complex, right? So what we have seen? We have seen the definition of enzymes. We have seen the some history in a short, because this is your GPAT lecture, not your semester lecture. Okay. So whatever is important for GPAT, so I have extracted these things and I am going to explain you these things. Okay. So यहाँ पे what they do is they decreases the energy of activation. इसके ऊपर एक क्वेश्चन आ चुका है. Hollow enzyme is an active part. Hollow enzyme is an active enzyme composed of a two part. First part is the apoenzyme which is non-protein part. इसके ऊपर क्वेश्चन आ चुका है. Coenzyme is non sorry, apoenzyme is protein part and coenzyme is non-protein part. Now if coenzyme is covalently bind to the apoenzyme, then term prosthetic group is used. Prosthetic group is used. So इसके ऊपर क्वेश्चन आ चुके हैं hollow enzyme में. Now functional unit of enzyme is known as a hollow enzyme. तो इसके ऊपर भी क्वेश्चन आ चुका है. Multiple form of same enzyme is known as a isoenzymes. Monomeric means made up of a single peptide. Oligomeric means made up of few peptides. Multi-enzyme complex is nothing but the having different enzymes. Right. Now we'll see the next part in this. Now classification of enzymes. Classification of enzymes. Right. So इसके ऊपर भी क्वेश्चन आ चुका है. Classes of enzyme as per the International Union of Biochemistry. So IUB as per the International Union of Biochemistry. As per the International Union of Biochemistry, IUB. Enzymes are classified into 6 classes. Okay, enzymes are classified into 6 classes. इसके ऊपर क्वेश्चन आ चुका है. There is one table in the Satyanarayana also, and most of the books also have this table. Okay, so first is nothing but the oxidoreductase. Class क्या है? Oxidoreductase. Now using this name you will understand what is the role of these enzymes. So oxidoreductase means they are engaged in the, or they are involved in the oxidation reduction reaction of your body. So in the particular metabolic cycle, so oxidoreductase enzyme का role क्या रहेगा? So they are involved in they are involved in the oxidation and reduction reaction. So what are the examples of oxidoreductase? So आपका alcohol dehydrogenase. So what is the role? Alcohol dehydrogenase will convert alcohol into aldehyde. So it is a glucose oxidation of alcohol. So oxidoreductase enzyme example is alcohol dehydrogenase, then cytochrome oxidase, you know, then amino acid oxidase, you know, all these are the examples of oxidoreductase. So what is the role of oxidoreductase? So role is nothing but the oxidation and reduction reaction. Oxidation reduction reaction. Oxidation reduction reaction. ऑक्सीडो रिडक्टेज इसका नाम में ही रखा है ना, आप बोलते हो नाम में क्या रखा है, नाम में बहुत कुछ रखा है। ऑक्सीडो रिडक्टेज मतलब those enzymes which catalyze the oxidation reaction and reduction reaction, है ना। now what is mean by oxidation reaction? So gaining of electrons known as oxidation and losing of electron known as a reduction, right? And same thing they were asked in the GPAT 21. So following category of enzyme, they are engaged in the reactions which involves the gaining and losing of electron. क्वेश्चन का कैसा था, क्वेश्चन ऐसा था, following category of enzyme are involved in the reaction which involves gaining and losing of electron. तो gaining and losing of electron मतलब क्या, ऑक्सीडेशन एंड रिडक्शन, है ना। सो ऐसा क्वेश्चन कंफ्यूज करने के लिए पूछा था, oxidoreductase, right? Now next one is the transferase. What do you mean by, what do you mean by transferase? So from that name you will understand some, you will get some idea. Transferase means what? Transferase means transfer of what? Certain groups. So what they do, they are involved in the transfer of groups from one molecule to another molecule. So what they do, they are involved in the transfer of groups from one molecule to another molecule. Example क्या है भाई इसका, है ना। example is nothing but the hexokinase, है ना, glucokinase. Hexokinase is also known as a glucokinase. So what did it do? Hexokinase क्या करता है, आपने देखा है glycolysis में. So what hexokinase will do? So glucose is converted into glucose 6 phosphate by hexokinase or also known as a glucokinase. So what that do? That transfer the phosphate group to the glucose. That's why glucose 6 phosphate. So transferase enzymes, they are in the transfer of group. Okay, like okay, hexokinase will transfer phosphate group. Transaminase, भाई transaminase में क्या कर सकता है, उसका नाम में लिखा है trans मतलब transfer, aminase मतलब amino group. So what transaminase will cause? It will do transfer of amino group NH2 group. So this transaminase mostly involved in the metabolism of protein and amino acids. Transmethylase, obviously transethylase. So what they will do, they will transfer ethyl group respectively. इन फॉस्फोराइलेज क्या करेगा, फॉस्फेट ग्रुप मॉलिक्यूल को ऐड कर देगा, है ना। सो ट्रांसफर ऑफ ग्रुप फ्रॉम वन मॉलिक्यूल टू अनदर। नेक्स्ट कैटेगरी, नेक्स्ट एंजाइम होता है आपका हाइड्रोलेज. सो हाइड्रोलेज विल डू, it will cause hydrolysis, it will cause hydrolysis of the molecule by adding the water molecule, it will cause hydrolysis of molecule by adding H2O molecule. Okay, so for example is nothing but the lipase. What lipase will do, it will hydrolyze the lipid को hydrolyze करने वाला एंजाइम होता है lipase. कोलिनेस्टरेज मतलब क्या, एसिटिल कोलिन को हाइड्रोलाइज करने वाला कौन है, कोलिनेस्टरेज. ओके, एसिटिल कोलिन को हाइड्रोलाइज करने वाला एंजाइम क्या है, कोलिनेस्टरेज, राइट. नेक्स्ट क्या होता है, अल्कलाइन फॉस्फेटेज, पेप्सिन, है ना, पेप्सिन है, यूरेज है, यूरेज एंजाइम होता है ना, यूरेज, उसका काम क्या होता है, इट विल ब्रेक डाउन द यूरिया का हाइड्रोलाइसिस करेगा, है ना। ये यूरेज एंजाइम सा बहुत खतरनाक होता है, किसमें होता है ये होता है आपका एच पाइलोरी. एच पाइलोरी, एच पाइलोरी से क्या होता है, एच पाइलोरी से आपको मिलता है, यस एनीबॉडी, एच पाइलोरी इज अ पॉजिटिव ऑर्गेनिज्म फॉर व्हाट, एनीबॉडी, एच पाइलोरी से क्या होता है आपको, yes, it causes the ulcer, peptic ulcer, duodenal ulcer. So 99% of duodenal ulcer they are caused by this H pylori. 99% duodenal ulcer they are caused by H pylori. Remember this H pylori, H pylori, H pylori means Helicobacter pylori is the organism bacteria, okay, which is responsible for the duodenal ulcer. So 99% of duodenal ulcers they are caused by H pylori. तो इसके ऊपर भी एक क्वेश्चन आ चुका है. Now why this H pylori survives in the acid environment? आपके स्टमक में तो एसिड है ना. आप अगर बाहर कोई स्ट्रीट फूड खाते हो, समोसा खाते हो, जलेबी खाते हो, या फिर पानी पूरी खाते हो, लड़की स्पेशली, है ना, तो वो पानी पूरी जलेबी जो भी समोसा हो रहा है, आजकल सैंडविच हो रहे हैं, तो उसमें क्या होता है कि उसमें कंटेमिनेशन होता है उसमें. सो मोस्ट ऑफ द फूड दे आर कंटेमिनेटेड विद ई कोलाई एंड साल्मोनेला. तो उसमें से ई कोलाई होता है, साल्मोनेला होता है बैक्टीरिया, और दिखते नहीं है आपके आंखों से, लेकिन होता है, आप खा लेते हो उसको, है ना, बट आपको कोई दिक्कत नहीं होता. सो यू विल नेवर हैव इन्फेक्शन. व्हाई? बिकॉज यू हैव एसिड इन योर स्टमक एंड in the acid most of bacteria they are destroyed. Okay, but obviously if food is okay, if in the food is contaminated a lot, है ना, अगर स्ट्रीट फूड में अगर कंटेमिनेशन बहुत ज्यादा है तो कभी भी क्या होता है, आपको डायरिया हो जाता है कुछ खाने से. व्हाई? बिकॉज दैट फूड हैव लॉट ऑफ है ना कंटेमिनेशन, दैट्स व्हाई, है ना. देन व्हाई दिस एच पाइलोरी कैन कॉज ड्यूडेनल अल्सर। इसके पीछे भी एक रीजन होता है कि एच पाइलोरी है ना, बाकी जितने भी बैक्टीरिया वगैरह होते स्टमक में जाने के बाद मरते हैं, तो ये एच पाइलोरी साला क्यों जिंदा रहता है ना, और जिंदा रहता है और क्या करता है, ड्यूडेनल अल्सर क्यों करता है, क्योंकि उसमें एक एंजाइम रहता है, कौन सा, यूरेज नाम का एंजाइम। तो यूरेज नाम का एंजाइम जो है वो क्या करता है, यूरिया को ब्रेक डाउन करता है। यूरिया मतलब क्या है, NH2 डबल बॉन्ड O NH2। अगर यूरिया का ब्रेकडाउन हो गया तो क्या रिलीज होगा, ऑब्वियसली क्या रिलीज होगा, अमोनिया। तो अमोनिया क्या है भाई, एसिडिक बेसिक, तो बेसिक है ना। तो अमोनिया जब रिलीज होता है ना, यूरियस अमोनिया रिलीज करता है कहाँ पे, बैक्टीरिया के सराउंडिंग में, है ना। बैक्टीरिया का सराउंडिंग मतलब बैक्टीरिया वैसे भी टाइनी होता है, बहुत छोटा होता है, एंड surrounding area of bacteria is also very small, so it will release ammonia in the surrounding of bacteria. That ammonia will neutralize the acid, and that's why this H. pylori or Helicobacter pylori is able to survive in the stomach and can cause peptic ulcer and duodenal ulcers. Are you getting or not? Okay, so hydrolysis, next class क्या होगा आपका, lysis. Lysis, okay, lysis, what will they do, lysis, they are involved in the addition and elimination reaction. So addition reaction, elimination reaction, so addition of H2O, ammonia, carbon dioxide. Okay, so addition and elimination reaction. So aldo, okay, example is nothing but the aldolase, aldolase, fumarase, histidase, all these are the example of lyases. Now, what will next class is nothing but the isomerase. Isomerase. So, what isomerase will do? Obviously, in the name, isomerization reaction. So, what will it cause? Isomerization reaction. है ना, D का L बनाएगा, L का D बनाएगा, या फिर आपका for example जैसे we have seen in the glycolysis. Okay, so there is a formation of glycerol 3 phosphate, and there is a formation of dihydroxyacetone phosphate. Okay, so from the fructose 1,6 bisphosphate molecule, so that is known as a splitting phase. तो जो glycerol 3 phosphate बनाए और dihydroxyacetone बनाए, तो वो क्या है, सेम कार्बन का है, उसमें फर्क क्या है, ऐसे ये कीटोज है और एक अल्डोज है ना। तो उसके बीच में एंजाइम लगता है, कौन सा, आइसोमरेज। तो क्या करता है, डायहाइड्रोक्सी कीटोन जो होता है, डायहाइड्रोक्सी उसको ग्लिसराइड 3 फॉस्फेट में कन्वर्ट करता है, सो आइसोमराइज। Okay, so isomerase enzyme, right? Okay, so you can see phosphoexose isomerase, then glucose isomerase, etc., etc. Then ligase enzyme is there. Next is the ligase. Ligase means what? Two ligate, two ligate or two ligate. Two ligate means two ligate मतलब क्या? Two tie. Two ligate मतलब to tie. To bind. Okay, so to bind or to bind, to tie. Tie up करना, अपन बोलते हैं कि हमारे कॉलेज ने इसके साथ में tie up किया, सो मतलब बॉन्ड हो गए, बॉन्ड हो गए उसमें। लाइगेज मतलब क्या है, सो it is involved in the synthesis, it is involved in the condensation reaction. So two molecules, it will be tied up together by this ligase molecule, okay, or ligase enzyme. So what is the example, glutamine synthase and succinate thiokinase, so etc. So what they are involved, they are involved in the condensation reaction which are usually ATP dependent. तो ये चार्ट आपको बाय हार्ट होना चाहिए क्योंकि क्वेश्चन इसके ऊपर ऑलरेडी आ चुके हैं, राइट। सो यू कैन रिमेंबर द क्लासेस लाइक initial letters of this class. Okay, so O for O, then T, O T H L I H L I L I L, सो याद कैसे रखना है, ओ टी एच एल आई एल, उसका एक टर्म बनता है ओथ लील, ओथ लील, ओ टी एच, यहाँ पे लिखता हूं मैं ओ टी एच, ओथ। ओथ मतलब क्या होता है, शपथ, ओथ लील, ओथ लील, ओथ लील एल आई एल, ओथ लील, ओ टी एच एल आई एल ना। ओथ लील मतलब ओथ ली है ना, अपन ऐसे बोलते ना, मैंने शपथ ली, तो वैसे ओथ ली, तो ली की जगह लील बोलो, ओथ लील है ना, बच्चे जैसे बोलते हैं लील है ना, ओथ लील। तो ओथ लील यू कैन रिमेंबर दिस 6 एंजाइम्स, सो ऑक्सीडेटिव, ट्रांसफरेज, हाइड्रोलाइजेस, लाइजेस, आइसोमरेजेस एंड लिगेजेस। ओके, नाउ नेक्स्ट विल सी, बिकॉज तो इसके ऊपर ऑलरेडी एक बार क्वेश्चन आ चुका है, और भी क्वेश्चन आ सकते हैं, तो आपको याद रखना है। नाउ व्हाट एंजाइम्स विल डू, what enzymes will do, इसके ऊपर क्वेश्चन ऑलरेडी आ चुका है, they will decrease the activation energy, they will decrease the activation energy. So same thing is explained over here in this diagram, so you can see, okay, This is nothing but the time on x axis and energy on y axis. Okay? Energy on y axis. Now this one is known as a reactant or this one is known as a substrate. Okay? Reactant कोई enzyme catalyzed reaction में बोलते हैं अपन substrate. So this one is a substrate molecule. Now substrate is converted into product. Substrate is converted into product. This one is a product. Okay? Now in absence of enzyme, अगर enzyme नहीं है, in absence of enzyme, reaction kinetics without enzyme, in absence of enzyme, so it needs a more energy. It needs a more energy for collision. Okay, it need a more energy for collision. That you can see energy. यहाँ तक जाता है energy. Now in presence of enzyme, अगर enzyme है, so this energy is only up to this point B. अगर एंजाइम नहीं है तो इतना नहीं लगता है. से फॉर एग्जांपल यहाँ पे कोई वैल्यू लिखो आप 10 किलो कैलोरी लग रहा है अगर एंजाइम नहीं है तो रिएक्शन के लिए. अगर एंजाइम है तो सिर्फ 3 कैलोरीज लग रहा है. 3. That means the difference. So what enzyme will do? It will decrease the energy for activation. Energy for activation of reaction. Are you getting or not? क्वेश्चन ऑलरेडी उसके ऊपर आ चुका है. Factors affecting on the enzyme activity. There are lot of factors which affect the enzyme activity. So but only important I have listed and I'm going to discuss that only. Okay. So different factors like for example concentration of reactant is there, then time of reaction is there. Okay, then temperature is there, then pH is there, then enzyme concentration is there, then product concentration of product is there. Okay, so then okay, so these are the different factors. Okay, factors affecting on the enzyme activity. तो enzyme क्या करता है, एंजाइम क्या करेगा, जो रिएक्टेंट है जो सबस्ट्रेट है, सबस्ट्रेट का कन्वर्जन किस में करेगा, प्रोडक्ट में करेगा. सबस्ट्रेट विल बी कन्वर्टेड इनटू प्रोडक्ट बाय द एंजाइम. बट व्हाट आर द डिफरेंट फैक्टर्स व्हिच अफेक्ट दैट एंजाइम कैटलाइज्ड रिएक्शन. So how enzyme catalyzed reaction will be affected by different factors. So in that you are going to have temperature, reaction time, concentration of substrate, concentration of product, okay, concentration of enzyme also, okay, time, okay, lot of factors are there, co-factors and all that. So out of that only important factors we would like to see. Then first one is the concentration of enzyme. अगर enzyme का concentration कम ज़्यादा हो गया तो what is the effect on the enzyme catalyzed reaction, rate of enzyme catalyzed reaction. As the concentration of enzyme is increased, so rate of enzyme catalyzed reaction it will also increase proportionally. Proportional increase in the velocity of reaction or rate of reaction. तो enzyme का concentration बढ़ गया तो rate of reaction भी बढ़ जाएगा. Concentration of substrate का concentration increase in the velocity of reaction with within a limited range if you increase the substrate level, है ना. अगर substrate का concentration बढ़ा दिया आपने तो कुछ टाइम के लिए रिएक्शन का रेट बढ़ जाएगा, कुछ टाइम के लिए, लिमिटेड टाइम, बाद में नहीं. So concentration of substrate, increasing the concentration of substrate will increase the reaction rate or velocity of enzyme catalyzed reaction initially only for limited period. Then okay, order of reaction. So order of reaction. So you know that if the rate of reaction depends on the concentration of one reactant, it is known as a first order reaction. If the rate of reaction does not depends on the any one of the reactant, known as a zero order reaction. Rate of reaction depends on the concentration of two reactant, it is known as a second order reaction, है ना. तो अपने को ये ऑलरेडी आपको मालूम होगा. फर्स्ट ऑर्डर, ओके, ऑर्डर ऑफ रिएक्शन, फर्स्ट ऑर्डर विद रिस्पेक्ट टू द सबस्ट्रेट. Okay, first order reaction with respect to the substrate means there is a change in the velocity which is dependent on the concentration of single reacting component or substrate. So velocity of reaction is almost proportional to the substrate concentration up to the okay, substrate concentration, that is a less than km. km क्या होता है अपन देखेंगे. Okay, so in the first order, okay, first order with respect to the substrate will, okay, velocity of reaction is almost proportional to the substrate concentration, okay, and which is less than km. Velocity is less than km. Km क्या होता है, Michaelis Menten कांस्टेंट होता है. आई विल एक्सप्लेन. ये आपको आएगा. If the substrate concentration is much greater than the Km, the rate of reaction is independent on the substrate, and then it is called as zero order. When substrate concentration is less than Km, Km क्या होता है? Michaelis Menten constant, a constant for enzyme catalyzed reaction. I will explain that Km term later on. But when there will be first order reaction in case of enzyme catalyzed reaction, so only when the substrate concentration is less than Km, and at that time the velocity of reaction, enzyme catalyzed reaction, is almost proportional to the substrate concentration. The condition is only the substrate concentration should be less than Km value. Now if substrate concentration is greater than Km value, then the velocity of reaction is independent on the substrate concentration and it is a zero order. ठीक है. तो फर्स्ट ऑर्डर एंजाइम रिएक्शन कब होगा? जब सबस्ट्रेट कॉन्सेंट्रेशन क्या होगा? कम होगा. किससे? केएम से. केएम क्या होता है अपन देखेंगे. यस. नाउ इफ अगर सबस्ट्रेट कॉन्सेंट्रेशन ज्यादा है, बहुत ज्यादा केएम से, तो उसके जीरो ऑर्डर काइनेटिक्स फॉलो करेगा एंजाइम. ओके, जीरो ऑर्डर. व्हाट इज मीन बाय enzyme catalyzed reaction? So enzyme catalyzed reaction means what? Enzyme catalyzed reaction क्या होता है कि enzyme catalyzed reaction मतलब क्या होता है? आपका enzyme, यहाँ पे क्या रहेगा? substrate. मतलब आपका reactant plus, okay, plus enzyme. Substrate plus enzyme. What will you get? You will get a product. You will get a product. So it is very simple, but if it is in a two step, okay, अगर दो स्टेप में होता है, so फॉरवर्ड रिएक्शन होता है, ओके, एंड रिवर्स रिएक्शन भी होता है, फॉरवर्ड एंड बैकवर्ड रिएक्शन. ओके, सो डिपेंडिंग ऑन दैट, इफ यू टेक, ओके, फॉरवर्ड रेट कांस्टेंट k1, ओके, then backward rate constant k2 and rate of formation of product k3, so then the Michaelis Menten constant km is equal to k1 is equal to k2 plus k3 divided by k1. कुछ ऐसा आपका यहाँ पे आएगा. So what is km? Km is nothing but the, okay, km is nothing but the, it is known as the Michaelis Menten constant or Briggs and Haldane's constant. Briggs and Haldane constant. Briggs and Haldane constant. K2 plus K3 divided by K1. Now this enzyme catalyzed reaction. एक enzyme catalyzed reaction में क्या होता है? So what happens? Enzyme plus substrate. Enzyme plus substrate. ये combine होके क्या बनेगा? Enzyme substrate complex. Enzyme substrate complex. The rate at which enzyme substrate complex is formed. So suppose that constant क्या है? उसका rate constant K1 है. Now ये क्या है? Reversible reaction होता है. So forward reaction constant is K1, backward reaction constant is K2. From ES complex is there. That means enzyme substrate complex, and finally product is formed. Okay. Finally product is formed. Now for this reaction K3 is the constant. अगर ऐसा आपने किया, so final rate constant will be given by K2 plus K3 divided by K1. This is known as a Briggs and Haldane's constant, and also called as a Michaelis Menten constant. Okay. Michaelis Menten constant. Now Michaelis Menten equation. So all these three, okay, Michaelis Menten equation. There is one more equation known as a Line-Weierbuch double reciprocal plot. So they are the related with the substrate concentration. What is the effect of substrate concentration on the enzyme catalyzed reaction? इस substrate concentration का क्या effect होता है? कि अगर substrate concentration बढ़ गया, कम हो गया, तो उसका क्या effect है कि enzyme catalyzed reaction जो है, उसके ऊपर क्या effect है? वो उसके related क्या है? Michaelis Menten equation है और Line-Weierbuch double reciprocal plot है. तो Michaelis Menten equation क्या है कि V is equal to Vmax into substrate concentration divided by Km plus S. क्या इक्वेशन याद रखना है आपको? V is equal to Vmax into substrate. Vmax क्या है? V is nothing but the velocity of enzyme catalyzed reaction. Okay? V is the velocity of enzyme catalyzed reaction, means rate of enzyme catalyzed reaction. ये rate होता है. Vmax क्या है? It is the maximum velocity, okay, of the enzyme in the enzyme catalyzed reaction. Maximum. What is your substrate concentration? Okay, what is Km? Km is known as a Michaelis Menten constant. Michaelis Menten constant, or also known as a Briggs Haldane's constant. Okay, Michaelis Menten constant. What is that? I will explain. Okay. So what is Michaelis-Menten constant? It is a concentration of substrate. Okay, concentration of substrate at which there is a half of the maximum velocity in the enzyme catalyzed reaction. So that is known as the Michaelis-Menten equation, Michaelis-Menten constant. Okay. Now ये equation ऐसा होता है V is equal to Vmax into S divided by Km plus S. क्योंकि इसके ऊपर क्वेश्चन आ चुके हैं इसलिए मैं यहाँ पे आपको दो दो तीन तीन बार बोल रहा हूँ. Michaelis-Menten equation V is equal to Vmax into S divided by Km plus S. So what is Km? Michaelis-Menten constant. Where यहाँ पे देखो V is the measured velocity, Vmax is the maximum velocity, S is the substrate concentration, and Km is the Michaelis-Menten constant. Okay, so what is that? The substrate concentration to produce half maximum velocity, half of the maximum velocity. The substrate concentration to produce half of the maximum velocity, half of the maximum velocity. अगर इसको सॉल्व करेंगे तो सॉल्व ये इक्वेशन का पूरा सॉल्व जो करेंगे उसका भी दिया है बुक में, आप जाके देख लो वहाँ पे. सो जैसे यहाँ पे Vmax इंटू एस डिवाइडेड बाय क्या है Km प्लस एस, मतलब Km प्लस एस सबको डिवाइड है. पहले Km से Vmax को डिवाइड करो, Km डिवाइड बाय Vmax इंटू एस डिवाइड बाय Km प्लस एस से सबको डिवाइड करो Vmax इंटू एस डिवाइड बाय एस बाय एस कैंसिल होगा. तो बाद में जब Km आएगा यहाँ पे एक यस रहेगा, यस इज इक्वल टू Vmax डिवाइड बाय Km है ना. तो Vmax क्या है हाफ है, अगर उसका हाफ से उसको सॉल्व करेंगे तो लास्ट में आता है सबस्ट्रेट इज इक्वल टू Km. ओके, लास्ट में एक इक्वेशन आता है, लास्ट में सॉल्व करके इक्वेशन अगर इफ यू सॉल्व दिस इक्वेशन ओके सो यू विल गेट अ सबस्ट्रेट इज इक्वल टू Km. इसको सॉल्व करेंगे हम Km. व्हाट इज इट? So it means the concentration of substrate to produce a half of the maximum velocity. That is known as a Michaelis-Menten constant. Okay, are you getting or not? So Michaelis-Menten equation and Michaelis-Menten constant, they are the important terms. Okay, very very important. Okay, if you if you plot a velocity versus substrate concentration, if you plot velocity versus substrate concentration, so you will get a curve. Okay, so you will get a first order kinetics. Okay, curve. What is okay? This is known as a Vmax, maximum velocity in the rate of in the enzyme catalyzed reaction, and this is known as a half of the maximum velocity. Now what is the Km? Km क्या है? It is a substrate concentration. देखो कहाँ पे Km एक substrate concentration to produce half of the maximum velocity, known as a Michaelis-Menten constant or known as a Briggs-Haldane constant. Same thing. तो आपको कंफ्यूज कर सकते हैं, रखना है आपको Briggs-Haldane constant and Michaelis-Menten constant. They are the same terms. Now by this Michaelis-Menten equation, so okay, or by this जो है ग्राफ, this graph is known as a Michaelis-Menten graph or Michaelis-Menten equation. So using this equation you cannot explain the effect of different inducers and inhibitors, on the enzyme catalyzed reaction. So that's why okay, so there is one more equation. First Km value क्या है? It is a constant characteristic of given enzyme. Km value जो है Michaelis-Menten equation में और enzyme catalyzed reaction में Km value is a important, it is a constant for particular enzyme. Okay, it is a constant for particular enzyme and it is not dependent on the velocity of reaction. It is not dependent on the concentration of substrate. Okay. Km value indicate the strong affinity between enzyme and substrate. What Km value indicate? Km value जो होता है वो indicate करता है affinity between substrate and enzyme. So how there is a affinity, how there is a collision between substrate and enzyme, that is indicated in the indicated by Km value. Right? Where okay, low Km value indicate the strong affinity. Now higher Km value indicate the higher Km value indicate the weak affinity between these two. Enzyme और substrate में कितना फास्ट उसमें इंटरेक्शन हो रहा है, वो कौन दिखाता है? अगर पर्टिकुलर रिएक्शन में Km वैल्यू कम है तो उसका मतलब ये होता है कि वहाँ पे एंजाइम एंड सब्स्ट्रेट का कोलिजन ज़्यादा हो रहा है, और जो जहाँ पे हाई Km वैल्यू है उसका मतलब ये होता है कि एंजाइम एंड सब्स्ट्रेट का कोलिजन क्या हो रहा है? कम हो रहा है. सो ए low Km value indicate the strong affinity between enzyme and substrate, whereas a higher Km value indicate the low affinity between substrate and enzyme. Okay, you have to be clear this, क्योंकि इसके ऊपर 100% क्वेश्चन. Most of the enzymes will have Km value in a range of 10 raised to minus 5 to 10 raised to 2. Most of the enzymes will have Km value in a range of this, and it is not dependent on the dependent on the Km value is not dependent on the concentration of enzyme. Km value is not dependent on the concentration of enzyme. Another plot is there. It is not another graph is there known as a line Bezier. Okay, line Bezier. Okay, line Bezier Burke double reciprocal plot. Line Bezier Burke double reciprocal plot. Okay, और लीन बियर बर्क डबल रेसिप्रोकल, जो भी आपको प्रोनाउंस करना है वो कर सकते हैं. लाइन बेवियर, line Beer Burke double reciprocal plot. Line Beer Burke double reciprocal plot. Okay. Now, as the name indicates, line Beer Burke double reciprocal plot. Reciprocal मतलब क्या है? Reciprocal को क्या बोलते हैं अपन? One upon जो भी है, one upon something. It is known as a reciprocal. So line Beer Burke double reciprocal plot. If you take a reciprocal of this above equation, अपन ने जो भी equation देखा है, इसमें Michaelis Menten equation है. So if you take the reciprocal of that equation, and if you solve that equation, you will get this linear line where Burke double reciprocal plot. Okay, there is a question on this on GPAT 2018. So if you take a, okay, इसका equation दिया था and identify करने के लिए हुआ था. So equation दिया था. So equation दिया था उन्होंने, वो 1 upon V max is equal to, है ना, उसके अपोजिट होता है वहाँ पे. ठीक है, एवरेज. सो 1 upon V max मतलब क्या है? इट इज 1 upon V मतलब क्या है? कि रेसिप्रोकल ऑफ वेलोसिटी ऑफ एंजाइम कैटालाइज्ड रिएक्शन. रेसिप्रोकल टर्म आ गया तो आपको फटक से यहाँ पे क्लिक होना है कि लाइन बेयर बर्क डबल रेसिप्रोकल प्लॉट, है ना. लाइन बेयर बर्क डबल रेसिप्रोकल प्लॉट. रेसिप्रोकल है 1 by है. Taking the reciprocal of Michaelis Menten plot, okay, Michaelis Menten plot, reciprocal of velocity, okay, taking the reciprocal of Michaelis Menten plot, then what happens? Reciprocal of velocity, there is 1 upon V versus the reciprocal of substance concentration. This gives a straight line. It gives a straight line with the slope. Slope क्या है उसका? Km divided by Vmax, and y intercept होता है उसका 1 by Vmax. Okay. Now this is known as the line viewer Burke double reciprocal plot. Line viewer Burke double reciprocal plot. Question already asked है इसका. So you can see over here. So if you take a reciprocal of, you know, last में ये equation आता है solve करने का. 1 upon Vmax is equal to Km divided by Vmax divided into 1 by S and 1 by plus 1 by Vmax. Okay. So line via Burke reciprocal plot, you can see over here. So it will get, you will get a straight line. If you plot a velocity versus substrate concentration, Km value कहाँ पे आएगा? Okay, Km value is minus, okay, minus, okay, 1 divided by Km. Then this is a half of Vmax. उसका y intercept क्या होता है and slope क्या होता है, इसके ऊपर क्वेश्चन आ चुके हैं. What is the slope? Slope क्या है? Y intercept कहाँ पे? So what is the slope? Slope is nothing but the 1 upon Vmax. The velocity, okay, slope Km divided by Vmax. Km divided by Vmax, and the y intercept is nothing but the 1 upon Vmax. Y intercept is nothing but the 1 upon Vmax. तो ये याद रखना है. Now this double reciprocal plot is useful in the understanding the effect of different inducers and inhibitors. Effect of different inducers and inhibitors. उसका ये यूज है. So we are discussing the concentration of substrate, effect of concentration of substrate on the enzyme catalyzed reaction. We have seen the Michaelis Menten equation. Michaelis Menten equation for first order. Now using Michaelis Menten equation, you will not get idea about the enzyme inhibitors and enzyme inducers. Okay, so that's why by taking a reciprocal of your Michaelis-Menten equation, there is one more graph known as a line where the Burke double reciprocal plot. Okay, so what here is there. Okay, so it is a reciprocal of velocity 1 upon v. ना पहले क्या था v is equal to ना v is equal to v max s divided by है ना km plus s. ऐसा था पहला ग्राफ माइकलिस मेंटेन इक्वेशन का इक्वेशन, राइट. वी आर सीन v is equal to Vmax S divided by Km plus S. ऐसा इक्वेशन किसका था माइकलिस मेंटेन इक्वेशन. तो उसका रेसिप्रोकल अगर करेंगे so you will get a line where Burke double reciprocal plot. Okay? So reciprocal of velocity 1 by v, that is versus reciprocal of substrate concentration. Reciprocal of velocity versus reciprocal of substrate concentration. If you plot this reciprocal plot करना है, पहले के में क्या था अपने सिंपली velocity versus substrate concentration प्लॉट किया था इन माइकलिस मेंटेन इक्वेशन में. वहां पे सिंपली वेलोसिटी वर्सेस सबसेट कॉन्सेंट्रेशन, वेलोसिटी था y एक्सिस पे, x एक्सिस पे क्या था सबसेट कॉन्सेंट्रेशन. फिर यहाँ पे क्या किया उसका रेसिप्रोकल लिया, मतलब उसको 1 बाय किया, 1 से डिवाइड किया. सो उसके डिनोमिनेटर को लेके नाउ दिस गिव्स अ स्ट्रेट लाइन, दिस गिव्स अ स्ट्रेट लाइन विद द स्लोप. उसका जो स्लोप है वो क्या रहेगा km डिवाइडेड बाय vmax एंड y इंटरसेप्ट होता है उसका 1 upon vmax. You can see over here. So line, okay, line where Burke double reciprocal plot. So यहाँ पे x एक्सिस पे क्या रहेगा सबसेट कॉन्सेंट्रेशन, y एक्सिस पे रहेगा वेलोसिटी. okay. So slope of this line where Burke reciprocal plot is nothing but the km divided by vmax. ये जो km divided by vmax, मैक्सिमम वेलोसिटी जितना रहेगा km माइकलिस मेंटेन इक्वेशन, सो माइकलिस मेंटेन कांस्टेंट उसको vmax से डिवाइड करने का. सो यू विल गेट अ स्लोप. स्लोप मतलब क्या उसका ये वाला स्लोप रहता है km डिवाइडेड बाय vmax, दैट विल गिव यू स्लोप. एंड y इंटरसेप्ट मतलब यहाँ पे y को कहाँ पे इंटरसेप्ट होता है 1 upon vmax इज नथिंग मतलब y इंटरसेप्ट, y एक्सिस को कहाँ पे क्रॉस कर रहा है 1 upon vmax पे. दैट इज नोन एज अ line where Burke double reciprocal plot. Are you getting अभी समझ में आया नहीं आया? If you read once book again, तुम एक बार आपने अगर बुक से पढ़ लिया ना तो ज्यादा समझ में आएगा आपको. बाकी कुछ नहीं इसमें. डबल रेसिप्रोकल प्लॉट मतलब क्या कि जो नॉर्मली के एम नॉर्मली अपना माइकलिस मेंटेन इक्वेशन है क्या है वेलोसिटी इज इक्वल टू वेलोसिटी इज इक्वल टू vmax into s है ना, vmax into substrate concentration डिवाइड बाय km plus substrate concentration. तो उसका इक्वेशन जो है वो है. और उसमें माइकलिस मेंटेन इक्वेशन में मतलब कैसे आया इफ यू प्लॉट वेलोसिटी वर्सेस सबसेट और सबसेट है ना, if you plot a substrate concentration versus velocity of reaction, that will give you some hyperbola type of curve, and that is nothing but the Michaelis Menten curve, hyperbola type of curve. Now if you plot a reciprocal of that, if you plot a 1 upon v, a reciprocal of enzyme is reciprocal of velocity against the reciprocal of substrate concentration, you will get a one line. This is the straight line which will have y-intercept known as a 1 upon vmax and slope km divided by vmax. Okay, km divided by vmax. That is known as a linear double reciprocal part. linear Burke double reciprocal part. इसके ऊपर भी क्वेश्चन आ चुका है. ये ऐसा इक्वेशन था, सेम इक्वेशन वाज देयर. ये इक्वेशन किसका है ऐसा पूछा था. ऑप्शन में था माइकलिस मेंटेन इक्वेशन और सिंपली है ना simply rate of reaction and line where Burke double reciprocal plot. So option was like this and answer was this line where Burke double reciprocal. You can go and check the paper. Now double reciprocal plot is useful in the understanding the effect of various inhibitors. Now, effect of temperature. Effect of temperature on the enzyme catalyzed reaction. Temperature अगर बढ़ाएगा तो obviously if there is increase in the temperature by particular degrees 10 degrees say for example 10 degrees 10 डिग्री से टेम्परेचर इंक्रीज किया रेट रिएक्शन क्या होगा ऑब्वियसली रिएक्शन का रेट बढ़ जाएगा क्योंकि इफ देयर इज इंक्रीज इन द टेम्परेचर देयर इज अ मोर कोलिजन बिटवीन टू मॉलिक्यूल्स देयर इज अ मोर इंटरेक्शन बिटवीन velocity of reaction increases with the increase in the temperature up to a maximum and then it will decline velocity of reaction catalyzed by enzyme इंक्रीजेज विद द इंक्रीज इन द टेम्परेचर, जैसे टेम्परेचर बढ़ोगे वैसे ही वेलोसिटी बढ़ेगा, है ना, कुछ मैक्सिमम वेलोसिटी तक जाएगा और बाद में कम हो जाएगा. वेलोसिटी ऑफ रिएक्शन इंक्रीजेज विद द इंक्रीज इन द टेम्परेचर अप टू maximum and then declines. You will get a bell shaped curve. Bell shape. You will get a bell shaped curve like this. You get a bell shaped curve. Okay. If we plot the temperature on x-axis and increase in the enzyme activity, मतलब reaction, velocity of catalyzed reaction. So you will get a this bell shaped curve. Initially increase the temperature will increase the rate of reaction. It will go up to a maximum rate of reaction, then it will decline. Okay, the temperature at which enzymes show maximum velocity of well, okay, the temperature at which enzyme will show maximum velocity in the enzyme catalyzed reaction, that is known as the optimum temperature of enzyme. इसको बोलते हैं अपन ऑप्टिमम टेम्परेचर ऑफ एंजाइम. तो आप देखो यहाँ पे बेल शिफ्ट कर्व मिलेगा आपको. इफ यू प्लॉट वेलोसिटी एंड टेम्परेचर, उसका अगर प्लॉट किया टेम्परेचर वर्सेस रेट ऑफ रिएक्शन, तो आपको एक बेल शेप कर्व मिलेगा. बेल शेप कर्व क्यों मिलता है? बिकॉज इनिशियली if you increase the temperature, rate of reaction will increase. Okay? It will go up to maximum rate of reaction will be here, and then it will again rate of reaction will decrease. Now why? Now the temperature, say यहाँ पे 48, temperature at which there is a maximum velocity of enzyme catalyzed reaction, it is known as the optimum it is known as the optimum temperature of your enzyme, because the optimum temperature means the temperature at which enzyme will show maximum velocity, maximum reaction rate, known as the optimum temperature, है ना. If temperature is increased, most of the enzymes will have optimum temperature in between 30 to 50, है ना. Most of the enzymes जो होते हैं उनका optimum temperature होता है 30 डिग्री सेल्सियस टू द 50 डिग्री. मतलब बॉडी का टेम्परेचर कितना होता है 37 डिग्री सेल्सियस. तो ऑब्वियसली, है ना, ऑब्वियसली बॉडी का टेम्परेचर ही 37 डिग्री सेल्सियस होता है ना. तो मोस्टली बॉडी में जो एंजाइम है उसका ऑप्टिमम टेम्परेचर भी उतना ही रहेगा. अगर उनका ऑप्टिमम टेम्परेचर 50-60, मतलब अगर टेम्परेचर आपका, इफ योर टेम्परेचर इज इंक्रीज, बॉडी टेम्परेचर इंक्रीज, अगर आपको फीवर है उस टाइम पे आपको क्या होता है, उस टाइम पे आपको वीकनेस होता है. उस टाइम पे आपके सब जो एंजाइम्स है बॉडी के, वो सब एंजाइम्स सप्रेस हो जाते हैं, बिकॉज उनका एक ऑप्टिमम टेम्परेचर होता है, उस टेम्परेचर के ऊपर ही वो काम कर सकते हैं ना, 37 डिग्री सेल्सियस. If your body temperature is increased, or if you increase the temperature too much in the enzyme catalyzed reaction, what will happen? There is a denaturation of enzyme, है ना. अगर बहुत ज़्यादा टेम्परेचर अगर इंक्रीज किया तो एंजाइम का डीनेचुरेशन होगा और वो रेट ऑफ रिएक्शन क्या होगा, कम होगा, है ना. So that's why at a optimum temperature, every enzyme has its optimum temperature. So most of the enzymes, they have optimum temperature between 37 to 50 degrees, 37 to 50. But there are certain enzymes, you know, there are certain enzymes which has a optimum temperature more than 50. जैसे फॉर एग्जांपल आपका पॉलीमरेज चेन रिएक्शन एक बायोटेक्नोलॉजी में आपको एक पार्ट है, उसमें पॉलीमरेज चेन रिएक्शन, उसमें एक एंजाइम लगता है आपको, टैग डीएनए पॉलीमरेज. ओके, सो दैट इज ऑब्टेन फ्रॉम थर्मस aquaticus bacteria, and that bacteria will survive at 90 degrees also. Why that bacteria will survive 90 degree? Because that has a enzymes which can survive at a 90 degrees. That's why the enzyme often formed that bacteria is known as tag DNA polymerase. Now tag DNA polymerase can have optimum temperature उसका होता है 76 डिग्री से कुछ ऐसा 76 डिग्री. मोस्ट ऑफ द एंजाइम का अपने बॉडी का ऑप्टिमम टेम्परेचर लेस देन 40 होता है ना, इवन से 30 to 50, है ना. तभी उस टाइम पे वो एक्टिव रहते हैं एंजाइम. अगर ज़्यादा टेम्परेचर हो गया तो वो एक्टिविटी नहीं दिखाई. ऑप्टिमम टेम्परेचर, राइट. नाउ टेम्परेचर कोएफिशिएंट मींस व्हाट? Temperature coefficient or Q10. Temperature coefficient is denoted by one term known as Q10. Q10 is nothing but the optimum temperature or temperature coefficient. Optimum temperature तो बोला मैंने. Temperature coefficient is nothing but the Q10 is defined as a increase in the velocity of enzyme. How much increase in the velocity of enzyme is there when you increase the temperature by 10 degree? अगर temperature 10 degree से बढ़ा दिया आपने तो कितना velocity increase होगा? So temperature coefficient Q10 is defined as a increase in the velocity of enzyme catalyzed reaction if you increase the temperature by 10 degree. Most of the enzyme will have Q10 between 2 to 40 degrees to 40. And there is one more interesting factor here. आप देखो, आप क्या करते हो, आपके सबके घर में फ्रिज होता है, फ्रिज, रेफ्रिजरेटर. रेफ्रिजरेटर में क्या करते हो आप, अगर कोई होटल से खाना ऑर्डर किया आपने, है ना, या फिर आपको अगर आज का बचा है खाना, कल उसको यूज करना है, तो आप क्या करते हो, उसको फ्रिज में रख देते हो। फ्रिज में रखते हैं तो वो खाना खराब नहीं होता। क्यों नहीं होता? उसका रीजन क्या है? उसका रीजन ही वही है, बिकॉज इफ बैक्टीरिया आर देयर इन योर फूड, अगर बैक्टीरिया का कंटामिनेशन होता ही है, एयर में भी बैक्टीरिया होती है, so if there is a contamination of food by bacteria, now bacteria also have, most of the bacteria will also have a enzymes, and that enzymes also have optimum temperature around 37-40 degree, okay, 37 or 40 degree उसके रेंज में होता है। if you decrease the temperature of your food, है ना, अगर आपने टेम्परेचर डिक्रीज किया 2 to 8 डिग्री सेल्सियस के ऊपर रख दिया आपने, कूल प्लेस में या कोल्ड प्लेस में, yeah, so what will happen? So this, though the bacteria are present in the food, but bacteria will not deteriorate the food, and bacteria उसको deteriorate नहीं कर सकते। क्यों नहीं कर सकते? क्योंकि उसको deteriorate करने के लिए बैक्टीरिया को वो जो फूड है उसको उसको यूज करना पड़ेगा, और यूज कब कर सकता है वो? उसके एंजाइम्स एक्टिव है तो, है ना। सो दैट्स व्हाई प्रिजर्वेशन ऑफ फूड इज डन बाय सब्जेक्टिंग द फूड इन अ कोल्ड स्लीव्स, है ना, आपको रेफ्रिजरेटर। तो वहां पे रेफ्रिजरेटर में खाना खराब क्यों नहीं होता है? उसका रीजन ये है, बिकॉज मोस्ट ऑफ द बैक्टीरिया विल हैव एंजाइम्स, एंड दैट एंजाइम इज आल्सो हैविंग ऑप्टिमम टेम्परेचर अराउंड 30 to 40। तो अगर आपने खाना कोल्ड प्लेस में रखा है तो वो खराब नहीं होगा। आर यू गेटिंग और नॉट? अगेन वन मोर इंटरेस्टिंग फैक्ट आई वुड लाइक टू शेयर विद यू। Suppose if there's if some patient is there whose operation, doctor has to do the operation, some surgical procedure they have to carry out, okay, and in that surgical procedure they need to suppress the metabolic rate of the patient। अगर मेटाबॉलिज्म उनको कम करना है वो पेशेंट का, और बाद में उनको ऑपरेशन करना है ना, कि मेटाबॉलिज्म नॉर्मल मेटाबॉलिज्म में वो ऑपरेशन नहीं कर सकते हो, तो वो क्या करते हैं मालूम है क्या? So they used to subject that they used to lower the body temperature of that patient। तो वो पेशेंट का बॉडी टेम्परेचर कम करेंगे पहले, है ना। अगर वो पेशेंट का बॉडी टेम्परेचर कम हो गया तो ऑब्वियसली उस पेशेंट में के जो एंजाइम्स है जिनका ऑप्टिमम टेम्परेचर होता है अराउंड 37 डिग्री सेल्सियस पोर्ट, है ना, 37 में, मोस्ट ऑफ द बॉडी के एंजाइम 37 ही के ऊपर ही, क्योंकि आपका बॉडी का टेम्परेचर ही 37 है, उनका टेम्परेचर है, तो बॉडी का टेम्परेचर कम किया तो ऑब्वियसली मेटाबॉलिज्म क्या होगा? कम होगा, क्योंकि वो एंजाइम क्या रहेंगे? एक्टिव नहीं रहेंगे। So your body's enzymes will be suppressed because of that temperature। So most of the time ऐसे surgical procedure they used to do by decreasing the body temperature।